The title for my talk goes as Nucleosome Reassembly During Transcription. And before I begin, I would like to thank the organizers for letting me present at such a short notice. So, okay, let me move a bit this side. So, before I begin, I would like to go over some basics. And this is what Rohit was mentioning earlier in his presentation. The length of the human DNA is in the order of 10 to the power 9 base pairs. If you arrange the DNA in single strands, it's near about in the order of 2 meters, whereas the size of the nucleus in microns. So DNA has to be compacted to near about 10 to the power 6-fold. And the way biological systems achieve this is by winding the DNA across these small protein complexes called histones. And these histones are then organized in higher order superstructures, which again arrange themselves into higher order superstructures, and its superstructures all the way down till you get chromosomes. So now that the DNA organization is there, we come to the next question that let's look at the structure of this thing. So here I have just represented this same cartoonish structure in more biological detail, but what you need to know is that the core complex around which the DNA is wound is composed of 8 protein subunits, and there are 2 of each of the 4 types, and this sort of gives rise to this bead on a string kind of nucleosome array, which sort of organizes themselves into higher order structures. The way this DNA is bound stably is by mostly charged interactions. So here the residues which are colored in blue are positively charged residues, and the DNA is more or less negatively charged, and these charge interactions stabilize this nucleosome. But there is a paradox here. So we are saying that the nucleosome has to be stable enough so that the DNA is strongly compacted. But we know that the DNA also has to be labile enough for something called polymerases. So polymerases are proteins which will take the DNA, make more DNA or RNA from it. So the DNA polymerase will create DNA from DNA, RNA polymerase is going to create RNA from DNA. So they have to be labile enough for the RNA polymerase or the DNA polymerase to pass through. So to put this into perspective, it's like you have a single lane road and you have 2 large 18-wheel trailers coming at each other and then somehow just passing through each other, because these are both order of kilodalton size complexes, and they have to accommodate each other on this track, which is the DNA. So how does one do it? So before we go into the specific details, I would like to say why keeping the nucleosomal identity is very, very critical in the life of an organism. So here is the added complexity. We just saw that there are these simple 8 units, but you can have variations of these 8 units, and each unit can have chemical signatures on their tails, which combinatorially can give rise to either expression of a gene or repression of a gene. And if a gene which is supposed to be expressed is either repressed, I mean, if genes which are supposed to be switched off are now switched on, or the vice versa, you are not going to have a good time. So keeping the nucleosomal identity is very required and very critical. So here is some biological jargon. Let's look over this. All in all, so what I'm going to present is some very recent work which was published in 2022 of some high resolution cryo-electron microscopy, which goes into detail of how the RNA polymerase in yeast, this is like a low order eukaryote, goes over through the DNA, histone bound DNA, and reassembles the nucleosome as the DNA is passing through. So again, taking a closer look at the histone octamer from a more structural perspective, you have the histone 3 and histone 4 dimer, and you have something called the H2A H2B dimer, which sort of organizes themselves in this manner, and the DNA is wound on this. And depending on the position of the DNA with respect to the entry site of the DNA, you can have superhelical sites. So the DNA is itself helical, and this thing is wound on, in a left-handed coil. So you have a superhelix, and you can have regions marked on the superhelix according to position, and it goes from its superhelical location -7 to superhelical position 0, and then to superhelical position 7, which is relative to this entry site called the dyad. Anyway, so now we will look at some of the high resolution cryo-EM structures, and the slide is visually noisy, but to summarize, this big chunk here is a multi-subunit complex, and this constitutes your RNA polymerase. The RNA polymerase is approaching this DNA wound around the nucleosome, and as it approaches, it has to push the nucleosome out. So you will see that compared to this, if you look here, the nucleosomes are more accessible, and the DNA has been sort of removed from the nucleosome. Now comes the interesting part, that how does one put the nucleosome back? So there is this protein complex which is known as the FACT or FACT complex, and it sort of stands for facilitates chromatin transcription. This is a tumor protein complex, it's a heterodimer, and the interesting part about this protein is it can bind DNA, it can bind the both tetramer and the dimer forms of histone 3, histone 4, histone 2A, histone 2B. So more or less this protein complex binds everything which is present in this nucleosome. So if something has to hold the entire organization together as the polymerase is passing through, it has to be this complex. And if you biologically in yeast cells reduce the amount of this complex, the cells are synthetic lethal. So basically the cells will die because this complex is critical for cellular function. So now let's look at these images. So the way to think about these images is you are looking at snapshots of a movie, and this polymerase is going over the DNA, and of course there are a whole bunch of names, but the names are not relevant. We'll focus more on the structure, and we'll focus more on the sort of what is the mechanically what is going on. So here this big polymerase complex is going through the nucleosome here. So this helical thing here is the DNA, and this extra thing here, which is the FACT complex, is binding the unwound histone complexes. So when the DNA is coiled around the histones, the FACT complex can't make contacts with this, so the polymerase has to be there. The unwinding has to happen so that the FACT complex can come and bind the DNA. Once the DNA has been bound, the complex is further stabilized by help from the polymerase itself. So the polymerase is a multi-subunit thing. It also contributes some of its protein domains to hold this structure in place. So now the polymerase moves further, and once most of the DNA has been unwound, the FACT complex can now completely contact the unwound histones, the naked histones basically. And now the critical part here is in these structures, what couldn't be found was that as the histones are removed and when they are reassembled, what is the sort of structure of the complex, but more or less the authors here sort of hypothesize that what's going to happen is based on some of the earlier biochemical studies, that once the polymerase has passed through, the nucleosome is going to reassemble slightly behind the polymerase as it passes by, and the polymerase, the histone subunits after the reassembly lacks some 1H2A/H2B dimer, which is then added back, and once this dimer has been added back, since the DNA is now completely wound, the FACT complex is no longer binding to it and it gets ejected. So the critical question here is that these large complexes are still staying in proximity to each other, and they have to somehow figure out the information that where this reassembly has to take place. And so in these sort of active systems, the question now becomes, what is the underlying mechanism that repeated transcription, which is taking place, sort of still leads to preservation of the chromosomal, the chromatin information in these systems. So to go ahead to do the final slide, as always, biology is not so simple. So in the same, within the same duration, there was another group which published a different set of structures. So this is from the previous ones which I was showing was yeast polymerase, RNA polymerase. These are from mammalian RNA polymerases. And here the picture is slightly simpler. What you have is the DNA being unwound, and the unwound DNA sort of folds back upon itself, folds back on the exposed histone, and again, so this is another simpler model, but which of these models is the actual picture remains to be closely examined and understood. And with that, thank you.
Thanks, Roni, for this nice talk.